Well, without further ado, it's my great pleasure to introduce our first keynote today, Dr. Chelsea Rochman, who's an assistant professor in ecology at the University of Toronto and scientific advisor to Ocean Conservancy. Chelsea received a PhD in ecology from a joint program between University of California, Davis, and San Diego State University in 2013. She was then a Smith postdoctoral fellow in conservation biology. She was hired as an assistant professor at the University of Toronto in the Department of Ecology and Evolutionary Biology in 2016. Chelsea has been researching the sources, sinks, and ecological impacts of plastic debris in marine and freshwater habitats for more than a decade. She's published dozens of scientific papers in respected journals and has led international working groups about plastic pollution. In addition to her research, Chelsea works to translate her science beyond academia. For example, Chelsea presented her work to the United Nations General Assembly and her work to the US State Department. Moreover, she has founded an outreach group called the U of T Trash Team, and more information can be found on her website, which is www.rochmanlab.com. Well, thank you so much for joining us, Chelsea. All the way from Toronto. So it's 7 p.m. over for where she's joining us from.
Thank you, Brad. Let me know if you can't hear me, and also let me know if you can't see when I share my screen.
So we can hear you. Yeah.
Oh, I think you have to, oh, there we go. Can you see my screen okay?
Yeah, sure can. I can hear you fine.
Okay, wonderful. Thank you for that kind introduction, and thank you for inviting me. It is 7 p.m. here, but that's a pretty reasonable hour, not too late. So what I want to talk about today are microplastics, which are an emerging contaminant, and it was actually really fun to hear Brad's introduction about describing kind of some of these properties of a lot of these emerging contaminants, because I want to talk about where microplastic fits and where microplastic maybe doesn't fit. So I'm going to frame it all in the multidimensionality of this contaminant and how that influences their fate and effects in aquatic ecosystems. So I'll start my talk with just a brief introduction to the contaminant microplastics. Then I'm going to talk a bit about how it is relevant to a persistent organic pollutant and talk about how that kind of relates to what we know about fate and effects, or hypotheses we may test about fate and effects, and then kind of moving forward with the research field. So I'll start with just a quick introduction to the contaminant. So I feel like a lot of people in this field start with this particular image, which some of you may have seen. It's a cover of Life magazine after World War II, and it sort of symbolizes the beginning of the plastic economy. So this magazine, when you open up the pages, goes to sort of celebrate the beginning of this new material that is flexible, it is durable, it is inexpensive, and it is convenient, or at least it can make lots of convenient items. Of course, it also makes items that we need, like medical equipment, and it makes planes and trains and cars lighter. This particular image, thinking about the single-use plastics that we all are familiar with, really brings to light kind of the celebration of the convenience of these items, which of course then led to sort of this inconvenient truth about plastic. So over the years, as we've increased production from about 500,000 metric tons per year to about nearly 400 million metric tons of plastic per year today, we produced a lot of this material. Some of it, so everything we've produced since 2015 is about 8,300 million metric tons. Some of it is still in use. I'm wearing plastic glasses on my face. My clothing is likely made out of polyester. And of course, the computer I'm speaking to you on has plastic in it. Some of this material, though, of course, becomes waste. Most of it becomes waste. A very small portion of that is recycled, less than 10%. Another small portion of that is incinerated. And the majority of it fits into a linear economy, either being discarded into a dump or a landfill, or potentially into the environment. And while we don't know exactly how much goes into landfill versus how much goes into the environment, we certainly know that a lot of plastic is getting into aquatic ecosystems as pollution. So just a few years ago, I was part of an international working group at SYNC, where we were trying to think about how much plastic waste enters aquatic ecosystems globally every year. And how will that change if we continue business as usual, do everything that we promised we would do, that's industry, government, and the people? And how hard do we have to work to get below 8 million metric tons, which is just simply the number that sort of brought this pollution to the global stage from Jenna Jambeck's paper in Science in 2015. So we estimated that we produce about, in 2020, let's say about 25 to 30 million metric tons, and that number may as much as triple or quadruple by 2030 if we continue business as usual. Now, I'm not going to talk today about this study or about what we found in terms of what we need to do in order to fix the problem and how it varies across the world. Instead, I just want to use it to talk about how much litter is going into the aquatic ecosystems and how that's really led to this global issue. So we knew in the beginning, we talked about plastic pollution, plastic debris in the middle of our oceans. This plastic here comes from the Great Pacific Garbage Patch, which was sort of brought to light in 2006. Of course, a lot of the debris is large pieces, but the reality of it is on a calm day, a lot of that plastic waste that enters into aquatic ecosystems breaks down into smaller and smaller pieces. As you can see here, pieces smaller than a pencil eraser, which eventually are and become microplastics. And so we know microplastics have become this global issue where plastic waste is degrading into smaller and smaller pieces. But we also know that microplastic is more than a plastic waste issue, in that in addition to plastic waste breaking down into these small particles, there are other sources of microplastics, such as microbeads from personal care products, pre-production plastic pellets that are made to produce plastic in the first place, washing our clothes, tires degrading as we drive, etc. And so this mixture of microplastics sort of leads to this kind of complex contaminant that we have out there. So one of the things I like to say are that microplastics are not microplastics are not microplastics, just like pesticides are not all the same. There are many different types of microplastics in the environment. They're made up of many different types of polymers. They have lots of different types of additives in them. They then are used to make many different products, which over time may break down into a size range that goes from the nano to 5 millimeters in size, in many, many different shapes, different colors, and they start to accumulate some of these contaminants over time when they end up in the environment. Some of these factors may tell us something about the source, where they came from, which is useful for mitigation. Some may tell us something about how they're transported or their pathways in the environment, and others may be relevant to toxicity. Another thing we've learned over the years is that microplastics are really becoming incredibly ubiquitous and persistent. Just like persistent organic pollutants, they're cycling in the atmosphere, they're cycling in the global dust cycle, they become part of the water cycle, and of course that means they're in more than just the oceans and freshwater, but also in our terrestrial ecosystems. When I first started researching this contaminant, it was us just trying to figure out where it was, how much was there, and maybe a little bit about the toxicity. Today, we understand a whole lot more. And in addition to really understanding how ubiquitous and pervasive this material is, we're also starting to learn something about risk, which I'll talk a bit about again at the end. But over the years, work that's been often led by Bart Coleman and his colleagues have tried to understand how we can put this microplastic in a risk assessment framework. And when we do, how that then tells us something about the risk we may see in the environment. So here you're looking at a paper on the left about freshwater and a paper on the right about marine systems, looking at basically taking a meta-analysis of all of the concentrations of microplastics that they see in freshwater and in the ocean, and overlapping that with basically creating a species sensitivity distribution from all the toxicity data out there to say, what is the concentration, in this case, the hazard concentration, where 5% of the species may be harmed or 95% of the species are protected. Similar thing in the ocean, where the more red here you see, the more risk you have. What this says is that there are some places in freshwater environments where risk is already occurring, and that's the same as in the ocean, and that if we continue business as usual, the amount of measurable risk that we see will increase. So that's a bit of background on this topic, this emerging contaminant, or maybe a contaminant that's already emerged. And now I'm going to talk a bit about how they may be related to persistent organic pollutants, which often are another group that becomes an emerging contaminant. So when I first started working on this topic, one of the people who introduced me to it, or whose work I found when I was searching online as an undergrad, was from Professor Shige Takata. And what Dr. Takata was interested in the beginning was not microplastic itself, but he was somebody who studied persistent organic pollutants, and he recognized that pre-production pellets that were common in the environment accumulated or worked like passive samplers to accumulate POPs. So he very eloquently asked people to simply mail him pellets that they found on the beach, say where they were from, and he would then measure the POPs on them to create maps such as these that show global distributions of, in this case, PCBs all around the world. Then he later started thinking about microplastics in and of itself as a contaminant, thinking about all the chemicals that were associated with them and how that may impact wildlife. It was a question I was really interested in as well when I first got into this field, was what are the chemicals that accumulate on plastics? What are the chemicals that are added during manufacturing? And how might those impact organisms? So during my PhD, I wrote a paper suggesting that maybe we should classify plastic waste in the environment as a hazardous substance if it was in an area where it would accumulate a lot of persistent organic pollutants, because POPs are hazardous substances. So kind of this idea of when you have a large accumulation of plastic in nature, you'll therefore have a large accumulation of POPs, and we can clean them out or get rid of them under polluter pays principles, the way we do hazardous substance. At the same time, other researchers were thinking about this kind of correlation between plastic and POPs, but saying, are microplastics themselves actually a persistent organic pollutant? And so that's what I want to talk about today, is are microplastics a persistent organic pollutant, or should they be? Or really, I don't need to answer that question for you today, but instead what I want to say is, what are the properties that are similar, and how can we use those properties to maybe think about what we know about plastics and maybe test hypotheses or create hypotheses about their fate and effects? So what does it mean to be a POP? A persistent organic pollutant is often ubiquitous, which I've already told you for microplastics. I'm not going to talk about that anymore. They are also often persistent, bioaccumulative, and toxic. So let's talk about these three things in relation to microplastic. Now, I don't tend to research the persistence of microplastics, so I'm not going to spend a whole lot of time on this particular aspect, but I can tell you that they are very persistent. They do break down over time by both a physical degradation, a chemical degradation, but also a biotic degradation. So we're starting to understand that eventually the building blocks of microplastics can become a carbon food source to wildlife. And actually what's been shown is more to microbial organisms. And then we also know that, of course, they break down over time with mechanical stress and sunlight and chemicals. So over time, as they break down, they tend to, this is when they fragment into microplastics. So instead of having a half-life where they sort of go away, over time, they tend to break up into more abundant, smaller pieces. But they do eventually assimilate and mineralize, but it takes a very, very long time. So they are incredibly persistent. This amount of time could be anywhere from hundreds to hundreds of thousands of years, a question we still don't really have a good handle on. Things I do tend to focus on in my research group is whether or not they are bioaccumulative and also whether or not they are toxic. So let's start with the question of bioaccumulation. We know that microplastics are inhaled and ingested by wildlife. There's lots of literature on microplastics in the gut content of animals. We also know that they can excrete. There's a body of literature about how long they're retained in the gut, as well as how much is excreted. We know that when they're in the gut content of an animal and they're eaten by a predator, that prey can transfer plastic pollution via trophic transfer to that predator. What we don't really know is whether or not they bioaccumulate or biomagnify. Now, some of you may be thinking, but we do know they bioaccumulate because we know that microplastics can translocate into the cells, in this case of a muscle, but also into tissue and into different organs. We see this in the literature. So they can translocate, and that's true. But the definition of bioaccumulations means they increase over time with the organism. So as an organism gets older and ages, the concentration tends to increase because it is accumulating in their tissues. And then when eaten by predators, they tend to biomagnify or increase up the food chain. So when I want to probe these questions, I want to probe them from not what's in the gut, but what's maybe actually accumulating beyond the gut. So for us, we've tried to ask questions in our lab to understand this. It's a work in progress. But I want to describe today a study that recently came out by Haley McElraith and Joel Kim in our lab that sort of were asking 3 research questions. One was, is there evidence of microplastic translocation in wild-caught fish? The second one is there evidence of bioaccumulation of microplastics? And third, do we see evidence for biomagnification, or do we maybe actually see trophic dilution? So for this first question, it's pretty simple. We simply just wanted to say, is there microplastics in the liver and the fillet of the wild-caught fish we were collecting from Lake Simcoe, which is in Ontario, here in Canada? So we expected it to be in the GI tract. Of course, we see that widely in Great Lakes fish. We also expected, if we saw translocation, that we would see more in the liver than in the fillet. The next question we really wanted to ask is, if we see translocation, do we see evidence for bioaccumulation? Meaning, do we see any evidence for an increase over time as a fish gets older? We didn't take otoliths, and these are wild-caught fish. So what we did instead was use fish mass or length as a proxy of age. So within a species, does the mass or length or the size of the fish positively correlate with the microplastics per gram of tissue? And if so, we were suggesting that may be an indicator for bioaccumulation. And then 3, for biomagnification, do we see a similar positive trend between trophic level of fish that we collected and the amount of microplastics in their tissue? So I'll start by showing you what we found in terms of the first question, which is, do microplastics, are they translocating beyond the gut? And we actually saw widespread evidence of microplastic translocation in wild-caught fish. For reference, this is smallmouth bass, largemouth bass, northern pike, yellow perch, brown bullhead, lake whitefish, and white sucker. This graph here shows you the larger pieces greater than 125 micron, and this graph shows you the smaller but not tiny pieces of 63 to 125 micron. The orange bars are how much was in the gut. We did not look in the gut in the smaller size fraction, since what we were really interested in was translocation, and we assumed it would happen more in the smaller fraction. The orange is the fillet and the purple is the liver. As you can see, we saw widespread translocation across all species, but we did not see more in the liver. We in fact found more in the fillet. So we did see translocation. Then we said, do we see evidence of bioaccumulation? So here we looked at how, again, the size of the fish on the x-axis compares to the number of particles in the fillet and liver on the fish on the y-axis. Here you see across all species, and I wouldn't focus there too much because really what we're interested in is within a species, how does it vary? So here you can see there's really no significant relationship. We do not see evidence of bioaccumulation. And any trend that you see, although again, not significant, tends to be negative, not positive, which suggests that potentially you're actually seeing growth dilution, meaning that as a fish grows and puts on more mass with the energy that it eats, it is putting on mass faster than it is translocating and accumulating microplastics. A question I got the other day is, do they excrete the microplastics? And I don't know. And for those of you that are in the room that may have researched nanoparticles, I'd be interested to hear about the excretion after translocation of nanoparticles, whether or not that's something we should explore here. For biomagnification, we also saw no evidence of biomagnification, no trend between trophic level and the number of microplastics beyond the gut. Now, again, this was kind of an opportunistic study, taking fish from the field. We didn't have huge sample sizes within each species. We continue to try to probe this question by sampling more fish in more contaminated areas in Lake Ontario, but also through different, more controlled experiments to try to look for these effects. Okay, so the next question is, are microplastics toxic? Do they tend to cause an effect at a high concentration? We know that there are fish out there are some animals out there that have what I would say relatively large amounts of microplastics being found in their bodies. We found a brown bullhead fish in Lake Ontario that had 915 particles in its gut. To me, that's a lot compared to what I've seen in the ocean. And Thomas Mays reported a shark with nearly 4,000 pieces. So the question is, does this lead to toxicity? And if it does, we might also ask, what are the mechanisms? Is it a physical effect, maybe a food dilution effect from eating microplastic and not its food? Maybe a physical toxic effect from the translocation of the particle, having a foreign item in your body, which can cause inflammation? Or is there a chemical effect from the chemicals associated with the plastic? In our lab, I would say we've seen evidence of both, which makes sense. We wouldn't necessarily expect it to be a clean story where it's only one or the other. So first, I'll show you some examples of evidence of a physical effect. One study led by a PhD student in our lab, Kennedy Bucci, and another led by another PhD student, Lisa Erdl, who's about to graduate in just a few months. So the first study was using fathead minnow, looking at how fathead minnow respond to both plastic in the tank when they can eat it and interact with it, but also plastic behind a screen where it's only allowed to leach the chemicals. Kennedy used both a low concentration and a high concentration for each treatment. And what she found was in the high treatment only, so the high concentration of the physical particles, a decrease in survival length and weight, and actually a significant increase in mortality from the ingestion and the interaction of these particles. So this suggests maybe a food dilution effect. Similarly, Lisa was interested in how microfibers may affect chironomids, so a midge in the lake, which actually are an important part of the food web, including for fathead minnow. So in this case, what she was interested in is, how do microfibers impact the midge? Does it matter whether it's a cotton microfiber or a polyester microfiber? Because a lot of people are interested in microplastic microfibers. And then, as we know, as a microfiber transfers into a lake or into an aquatic ecosystem, it likely goes through a wastewater treatment plant first. So she exposed both cotton and polyester microfibers to wastewater treatment plant effluent and then had clean ones and exposed those to the chironomid. What she found was it didn't matter whether it was cotton or polyester or had wastewater or not. The high concentration caused a delay in time to emergence for the chironomid, suggesting an effect to development. So again, potentially a physical effect causing maybe a food dilution effect. But we've also seen evidence of chemical effects, and these are from the types of microplastics where you might expect them to be a little bit more nasty chemically because of the additives and ingredients added to them. So in this case, both Anna Kola-Maheka and Leah Chibwe, who Leah is still a postdoc in the lab and Anna has moved on, we're looking at tire dust and the chemicals that leach from tire dust and how those impact fathead minnow. We found that the fathead minnow exposed to tire dust compared to those that are not tend to have this weird deformity where they're losing their pigment. They also have different, just other types of deformities, as well as changing in hatching success, as well as survival and heart rate. So what Leah really is trying to understand using non-targeted analytical chemistry is what chemicals are causing this. So can she actually pinpoint the chemicals more associated with the effects on the blue lines and see which ones tend to be less relevant to the effect? So she's currently doing TIEs in the lab to try to understand this. So it seems that plastics can cause kind of this physical effect and this chemical effect. And we also know from the literature that there's lots of evidence suggesting toxicity at many different levels of biological organization. So this includes those lower levels where you kind of better understand the mechanism of effect, to levels that may impact populations and individual organisms, to those more at the ecosystem level that will affect a community. And so over the years, I've tended to collaborate with different people to sort of create meta-analyses or synthesize all of the different effects that we see in the literature. So this started with the working group that I was in at NC's, and then it continued with Kennedy Bucci working on this as a chapter of her PhD. And now we've been doing this with the Southern California Coastal Water Research Project as part of a risk assessment, which I'll talk about at the end. But basically what we do is we go through the literature and we pull out any study that's tested a hypothesis about an effect, usually a laboratory test. And we mark the type of plastic, or sorry, the size of plastic that they were using in their experiment, or that was relevant to the effect, and the level of biological organization where it affected it. Over the years, the number of effects have increased, of course, because the body of literature increases, and we start to see more and more at these higher levels of biological organization. I'm going to ignore the macroplastic stuff right now, and for the purpose of this talk, just talk about the microplastic stuff. But of course, in addition to having these effects that are demonstrated, if we also look at the microplastic side, you can see that there's just as many, if not more, studies where they looked for an effect and did not detect one. And this makes sense. We know the dose makes the poison. But the other question I have is, when microplastic has all these different dimensions, right, the size and the shape and the polymer and the chemistry, it might be that it's not just the concentration that causes an effect. It might be because different traits that are associated with the plastics may cause different effects. So we've sort of tried to tease this apart, is how do these dimensions matter? So I'm going to show you now is a series of graphs that puts the concentration of microplastic on the y-axis and whether or not an effect was detected or not, either in the no's or the yeses. Here you can see that, of course, at some concentration, you start to get only yeses because it seems that there is a threshold for toxicity. The dose makes the poison, but you have lots of no's and yeses at the same concentration. And that's the interesting question, is why? We can start to pull that out a little bit more. We can say, does it have to do with the shape of the microplastic? Here you can see that for fibers, maybe there are more yeses at the similar concentrations than there are no's, suggesting that maybe microfibers are more toxic at lower concentrations than other types of plastics. We tried to probe this in other ways with the Southern California study, where we brought more literature into the mix, and it does seem that fibers may be more likely to cause an effect at a lower concentration than fragments or spheres. We can also see how it varies by polymer. I would say that at this time, even with increased literature, there's not a lot of evidence suggesting this in a strong way. But there are, of course, studies here and there that have really tried to understand the toxicity of different types. And so here you can see in Martin Wagner's group looking at PVC versus polyurethane versus PLA, where you do have differences in the toxicity between the microplastic types. And they also use a kaolin. So let's just say we use a natural particle. Do we see a difference between the toxicity between the natural clay particle and the plastic particles? And the answer that they see is yes. So there may be some sort of chemical that's associated with the polymer, of course, that may be causing toxicity. We've also tried to understand, in addition, how this varies by taxa, how it varies by level of biological organization, how it varies by the test duration, so if it's an acute test or a chronic test. But the last thing I want to focus on is size. And so here you can see that for the smallest size ranges, we only see a yes. And if we pull this out and look at it another way, you see more effects that are detected in the smaller size ranges, and this kind of pattern where you see less detected as it gets larger. Now, in the Southern California working group that we looked at, Scott Coffin dug into this a bit more, and it actually seems like it's not quite a linear trend, and that when it comes to a food dilution effect, larger particles are more likely to cause an effect at lower concentrations. But when it comes to kind of a translocation particle toxicity effect, it might be that you really need the high concentrations of those smaller particles, and you have more toxicity of the small particles as well as the large particles and something less in the middle. So the story may not be that simple, but it seems that size and definitely, well, not definitely size, potentially shape, and maybe even polymer type may matter. But when we look across the literature, most studies aren't really designing their experiments to ask that particular question. So it's hard to tease apart what traits of microplastics may lead to their effect, which I'll talk about in a little bit. So coming back to this idea of should microplastics be considered POPs? So let's go back to the specific traits that I talked about before. So first of all, are they persistent? Without a doubt, yes, they are. Are they bioaccumulative? It seems that maybe they're not, but I'm going to leave a question mark there because I think we still have some more work to do. And I'd like to continue to kind of focus on this question before I feel confident saying no. Are they toxic? Yes, there's evidence that microplastics can be toxic, and of course we know that they are ubiquitous. So yes, they have some similarities to POPs, but they're different. They are particles as opposed to chemicals. Of course, they're made up of chemicals, but they're physical particles. They come in many different shapes and sizes, and as they degrade over time, they break up, increasing in number as they decrease in size. They also have kind of this mixture of chemistries from both the polymers and the additives, and then of course they accumulate organic pollutants and metals as they're in the environment. So you may also think of them as kind of this multiple stressor, and every type of plastic out there has a different mixture of stressors. So they're complex. So what does that complexity mean then when we kind of think about how we move forward in this field? So before I kind of talk about that, just remind you about what some of these multi-dimensions are, these traits: polymer type, the additive chemistry, the size, the shape, the chemicals that accumulate on them, and maybe even the color. The color may matter for an organism that chooses to eat it, for example. The color also tells you something about source if you're interested in mitigation. So how do these traits affect the things that we know about fate and transport? So for persistent organic pollutants, we know that they tend to accumulate at that sea surface microlayer. We know that they move around with ocean currents. We know the same thing happens for microplastics. I don't think it's for the same reason. They move around with ocean currents, and some of them concentrate at that surface because of their density. So about half of all plastics that are made are positively buoyant, and about half are negatively buoyant, some being neutrally buoyant. And we know that the negatively buoyant sinks pretty rapidly, but the buoyant plastics will hang out at the surface and may sink with time as they foul and degrade. So similar with organic contaminants, they will move around the ocean in the same way. They will also sink down to the bottom and kind of get ingrained in the sediments. Similar again to organic contaminants, but for a different reason, this time due to the density. How they bury and then resuspend through the sediments, we're still working on understanding. One of the things I think that's really interesting is the atmospheric transport. I do not research this. I'm not an atmospheric chemist, but we do know that microplastics are transporting in global dust cycles. Some of this work has come to light through Dr. Janice Brani's work in Utah. She shows high concentrations of microplastics in remote areas in the air in national parks. Other work has also shown high concentrations in the Arctic, similar to persistent organic pollutants, where we have high concentrations of chemicals that are coming up from these dense urban populated areas at 60 degrees, following atmospheric cycles and coming down because they don't, in these cold temperatures, they're less likely to volatilize and they'll sink into the cold deserts. For microplastics, we see the same thing with high concentrations in the Arctic, but microplastics don't volatilize. Again, they are a physical particle. So one question I have is, when they rise around the equator, do they sink in the hot deserts as well? So if we were to go to the hot deserts and look for microplastics, would we see similar high concentrations the way we do in the Arctic? Again, this is not a topic that I research. It's just something that could be interesting for others to probe. The multidimensionality also makes how we measure them quite difficult. I think when we think about a risk assessment, we need to know the exposure or how much is in the environment. And then that's important for managers to think about whether or not they need to act. It's also important, most of this field started with just quantifying and characterizing microplastics. In the beginning and continuing through today, basically what happens is a student sits at a microscope for a very long time, quantifying and characterizing particle by particle. They give you the count of the microplastics in a sample, they characterize the shape and the color and the size, and using spectroscopy, you can get at an understanding of the types of polymers in your sample. Sometimes people do the whole sample. That takes a long time. Often people subsample. Some of this is starting to become automated, but most labs are still doing this in a very manual way. Now, people have tried to use pyrolysis GCMS to make it faster. In our lab, we can look at the mass concentration of plastics in a sample, but you lose something. You lose the count of particles, and if size matters, you don't know the size of the particles, and if shape matters, you don't know the shape of the particles. So there are definitely benefits to each type of measurement concentration or measurement of exposure, and I think they're complementary. But I was recently part of a working group in Southern California where we were asked to kind of come up with a way for how you would measure microplastics in drinking water and how you would use that information in a risk assessment. So we talked a lot about which one of these methods we should use. And if we build a risk assessment, whether it should be built using a mass concentration or using a count concentration. Those of us that have worked on other emerging contaminants are very used to a mass concentration. We're used to that always being used for our chemicals of interest. But for microplastics, it seems like the count may be more relevant, but this is still a conversation that I think is open to debate. So what we tried to do in California was actually create a multi-threshold risk assessment where different concentrations of microplastics suggested a different mitigation strategy. Having the highest threshold be the highest concern where you were asking the government to do more, potentially regulate, and of course the lower concentrations suggesting less and potentially eventually triggering a monitoring program. We of course also had to ask the question, in addition to, do we do this by count or do we do this by mass? Do we need to have a different risk assessment for the size, the shape, the polymer type, and the additive chemistry, which I told you a little bit about before. As we tried to expand on the meta-analysis and the synthesis of the literature, we created actually a Shiny app, which is, I don't think available yet, but soon to be available, but potentially it is already available. In any case, it's been created. Lots of function has been added to it. It is a database of all of the studies talking about a lot of the data I talked about earlier in terms of you can use it to say what concentrations tend to cause an effect. How does it vary by organism? How does it vary by the type of exposure you did? How does it vary by the shape of the material, the size of the material, etc.? And it also allows you to put it into a species sensitivity distribution. So we put all of the literature into this database, into this Shiny app, and we played with it to ask that first question of if we're going to build a risk assessment, do we use key particle characteristics, or can we just build one risk assessment kind of aligning all the particles together? So we evaluated, similar to what I showed you before, how much the size and the shape and the polymer type matter. And the consensus was that size is the only thing that really right now is very clear. And for the other questions, we really need more studies that again are designed to test that hypothesis. So what we said is that size matters. We know that food dilution, so the volume of the particles in the gut, can cause effects. That's shown in the literature. And we also know that small particles translocate, as I showed you before, and that those small particles can lead to maybe inflammation or other types of toxicity. So we decided that we would use two types of risk assessment and sort of have these two separate sets of thresholds developed for both food dilution and tissue translocation. But we did not say that the uniqueness of microplastics beyond size matters. So we actually used Bart Coleman's work for aligning particles to basically say, we're going to pretend that we're going to take the characteristics or the measurements of the plastics, kind of put them all into one shape and basically talk about them as more of a group of similar particles as opposed to the group of diverse particles. So then what we did is we used species sensitivity distribution to put all of the data in here to say, okay, can we create a risk assessment for microplastics? We were very careful about the data that we used. We had a QA procedure where we only used data that fit certain criteria. Did they have more than 3 concentrations that they used in order to fit more of a threshold? What were the different types of quality control that they used in order, like, for example, did they have controls? So there's a whole set of criteria. And then we've used them to set thresholds based on protecting either 95 or 90% of species, so an HD5 or an HD10. So this was sort of the framework that we created, where you had 4 different thresholds. One would lead you to investigative monitoring, one for discharge monitoring, one towards management planning, and one for more source control measures. Again, using the HC5 for most of them and the HD10 for the last. Using the first quartile or the median of the standard, the species sensitivity distribution line, and using either all of the endpoints or only some of the endpoints, the higher level endpoints. To give you a peek of what these thresholds were, here you can see them in particles per liter. One thing that may strike you is that these are incredibly low. 0.2 particles per liter is not a concentration unheard of in the environment. Whereas for the tissue translocation, the concentrations are a bit higher, maybe something you might expect. We then used kind of an IPC style, CC style voting to say how confident are we with both the framework and the thresholds. For the actual thresholds, it was a lower, and it was more to the like medium low. And this was because, again, these concentrations are quite low. I think my internet just cut out, so I apologize for that. Now, some people may be unsatisfied with this idea that we basically said, for this purpose, microplastics are microplastics except for size. Size matters, but we included a big size range. So we also, Kennedy and I kind of took some time to think about how might we actually try to include all of these different dimensions of microplastic. What if shape, polymer type, size, and the additives and the chemicals were important? Could we line them all up on a line and rank each particle within a sample based on where they fall on these different dimensions, and then sum them up to have a hazard quotient or a hazard number for each particle, sum those up within a sample and divide them by the volume of the sample to come up with a hazard number that's more relevant to both the diversity of the particles and also the concentration in the sample. So that was just a framework that we designed, but the reality is, as I said, a lot of studies are not designed to test those questions. There aren't very many studies that test one, say, for example, different polymer types while holding all of these other metrics constant. So more work is definitely needed, I think, to really come up with a risk assessment framework. And we need to kind of think about the complexity of this material and how that may lead us into that future, given that it is different than the frameworks that we've known and used for persistent organic pollutants. So I will stop there, and hopefully I still have time for questions. Thank you.